Welcome. In this session, we will continue to understand the transcription unit and the gene. So let's see about transcription unit and the gene. In the last session, I mentioned what is a cistron. So this cistron is a segment of DNA that codes for a polypeptide. So a segment of DNA that codes for a polypeptide. A segment of DNA that codes for a polypeptide is called cistron. And I told that the structural genes in a transcription unit, they can be monosystronic or polysystronic, right? So here what happens in case of eukaryotes is that only one gene that codes for a particular polypeptide will be transcribed onto a single mRNA, and that when translated will be producing only one polypeptide. So I say the monosystronic condition where only one polypeptide chain is synthesized from mRNA. Only one polypeptide chain is synthesized from mRNA. Whereas in case of polysystronic, right, more than one polypeptide chains are synthesized from a single mRNA, right? Of course, here also same, from a single mRNA molecule, right? Now such type of structural genes, we see this in eukaryotes. So monosystronic type we see in eukaryotes, whereas polysystronic we see in case of prokaryotes, right? We know that the DNA, through the process of transcription, it produces RNA molecules. Of course, here I'll be discussing with reference to messenger RNA. So here M stands for messenger, and we know this process is called transcription, right? And now this mRNA will be carrying information to synthesize one or more polypeptide chains. If it is in the eukaryotes, you will see that one mRNA will be producing only one polypeptide. Okay, that's what one mRNA will produce one polypeptide. So this is in eukaryotes. And when coming to prokaryotes, one mRNA will produce more than one polypeptide. So this condition we see in case of prokaryotes. Okay, of course, same process need to take place whether it is eukaryotes or prokaryotes, right? The transcription has to take place for the formation of mRNA molecule. So now this mRNA molecule, if it codes only for one polypeptide, then the condition is called monosystronic, right? And if we see that one mRNA molecule codes for more than one polypeptide, on more than one polypeptides, then the condition is called polysystronic. Now we will see how this polysystronic structural genes are present first. We'll see about polysystronic structural genes. Okay, so now here also same, the DNA, okay, in a process called transcription, it produces mRNA, and this mRNA will be having information to synthesize more than one polypeptide. So let's look at that structure of mRNA in prokaryotes. So structure of mRNA in prokaryotes. So right, and here say this is 5 dash and this is the 3 dash of mRNA in prokaryotes. So this region is a non-coding sequence. So this region is a non-coding sequence. So this is a non-coding sequence on the 5 dash, and similarly you will also see on the 3 dash end. So here also you will see some non-coding sequences. It means that these sequences will not be used in synthesis of polypeptide chain. So hence I call them as non-coding sequences. And then after that we see a start codon, right? And then the next stop codon. So here a codon is a sequence of 3 nitrogen bases, is a sequence of 3 nitrogen bases, right? That will be coding for a particular amino acid, say for example AUG. So in this, A stands for adenine, U stands for uracil, and G stands for guanine. So now this AUG will be coding for an amino acid called methionine. So later I will give you the entire details that what are all the codes and which code will be coding for which amino acid, that we will in detail study in the genetic code. Right, so start codon, which indicates that this is the start of that, okay, polypeptide chain, and this is the end of it. And then again say start codon here, and this is stop codon, and again here is start codon, and say this is the stop codon, right? So we have start codon, stop means this indicates this is the end of that, okay, polypeptide chain. Now from this point to this point there is one structural gene, and from start to stop again this is another structural gene. So this, say suppose this is structural gene 1, gene 2, and from here to here is also another structural gene, and that is 3, right? So now in this case this mRNA is going to code for 3 polypeptides. So in this case, okay, it is coding for 3 polypeptide chains, is coding for 3 polypeptide chains. Okay, that's the reason why we call it a polysystronic mRNA. So in case of prokaryotes, the mRNA is capable of synthesizing, so it can be used in synthesizing more than one polypeptide. So for that reason in prokaryotes the mRNAs are polysystronic, right? Now let's look at monosystronic. see about monosystronic structural gene. So monosystronic structural gene. So in this case, right, we will be seeing this in eukaryotes. We'll see in case of eukaryotes where DNA, of course, by the process of transcription, it results in the formation of mRNA, and this mRNA will be coding for only one polypeptide. So it codes for only one polypeptide. And moreover, okay, in case of eukaryotes, directly mRNA is not synthesized. So what actually happens is DNA first gives rise to hnRNA. So this HN stands for heterogeneous nuclear. So it's called heterogeneous nuclear RNA. This heterogeneous nuclear RNA will undergo a process called RNA splicing, and then finally it results in the formation of mRNA. So here hnRNA is an immature RNA molecule, right? Whereas mRNA is a mature molecule, right? So in case of eukaryotes, there will be an immature RNA formation first, that's called hnRNA. That hnRNA has to undergo a process called RNA splicing to form a mature RNA molecule. So such thing is not seen in case of prokaryotes. So in prokaryotes, there will be direct formation of mature RNA molecule, right? So now let's see about, okay, what's this hnRNA. So this hnRNA, the immature RNA molecule, it will be having introns and exons, right? So that's what actually is this intron and exon. Intron is a non-coding sequence. So it is a non-coding sequence, or also called, right, unexpressed sequences. Unexpressed sequences. So these introns do not code for any part of the polypeptide. So they are intervening sequences. Then coming to exons, the exons, okay, are coding sequences. Exons are coding sequences, or also called expressed sequences. Expressed sequences. So the exons will be coding for the polypeptide. So code for polypeptide, codes for polypeptide, whereas intron doesn't code for polypeptide. So it doesn't code for polypeptide. Okay, so intron is a non-coding sequence, and exon. So both are seen in case of this hnRNA. Now the point here is these introns should be removed, and only the exons should, okay, join to form a complete sequence that helps in the synthesis of a polypeptide chain. Okay, and that process is called RNA splicing. So what happens in RNA splicing is these introns are removed and exons are joined, because I told you introns, they do not carry any information for the synthesis of polypeptide, hence they need to remove. Now such condition we don't see in case of prokaryotes. So I did not discuss anything in prokaryotes. I am discussing okay, in case of eukaryotes only. That's the difference between the prokaryotes and eukaryotes. There mRNA will be directly formed, but in case of eukaryotes mRNA is not directly formed. So first there will be an immature mRNA, that is called hnRNA, which will be having these introns, exons, and there are so many other things that are not seen in case of this hnRNA. And after all that additions, deletions, that finally it results in the formation of mature RNA molecule. Right, now let's look at these introns and okay, exons, how they are. So when hnRNA is formed, say suppose this is the hnRNA, you may see here intron, then exon, intron, and then exon. Okay, and now during RNA splicing, what happens is all those introns are removed, only exons joined. So only these exons are joined. In eukaryotes, that's what we see, introns are interrupting sequences which are present, okay, between these exons. So for that reason, right, the eukaryotic genes are called split genes. Eukaryotic genes are called split genes, and okay, presence of these introns, exons is what actually is called split gene arrangement. It's called split gene arrangement. And after RNA splicing, right, all these introns are removed, only exons will be joined. Okay, says this is intron 1, intron 2, exon 1, exon 2, right? So in this case introns are removed, right? So that's what the eukaryotic genes are called split genes, and such split genes we don't see in case of prokaryotic organisms, right? Now let's look at the structure of mature mRNA in eukaryotes. The structure of mature mRNA in eukaryotes, right? So I told you already, hnRNA is an immature one that will be having introns and absence of some other additions that we'll be discussing now, right? So there must be some additions and deletions. Once that is finished, it results in the formation of mature mRNA molecule. So now, so say this is 5' end of mRNA and this is 3' end of mRNA molecule. And just like how we have seen in case of prokaryotes, here also we will see a portion that will be not translated, and this is called 5' UTR, will contain non-coding sequences. So this region will be having non-coding sequences, right? So here UTR, that stands for untranslated region, stands for untranslated region. Similarly, on the third end also you will find same non-coding sequences. It's called 3' UTR, right, untranslated region. And now, right, after that we find here the start codon, and then here we have is stop codon, and from one start codon to stop codon, right, is the structural gene. So this is the structural gene, and now this structural gene will be having only exons. There will be no introns. So structural gene with only exons. Now on this fifth end there will be addition of methyl, okay, guanosine triphosphate. So there will be okay addition of right, it's called methyl guanosine triphosphate. So it's called MGPP, that stands for methyl guanosine triphosphate. It's called methyl guanosine triphosphate. And in the similar way, on the third end, there will be addition of lot many adenylate residues. So these are which form something called poly A tail. So the meaning of poly here A tail is adenylate residues. So they are all adenylate residues. Okay, so many adenylate residues, of course, the number will be around 200 to 300 such adenylate residues will be added on the third end. So such additions we don't see in case of mRNA of prokaryotes, right? So this is the final structure of mRNA molecule, and you can see here only one structural gene. So this whole mRNA is going to code for only one polypeptide, right? So this structural gene will be coding only for, so codes only for one polypeptide, codes only for one polypeptide, right? Whereas in case of prokaryotes, we have seen that codes for more than one polypeptide. So here there are additions like methyl guanosine triphosphate and poly A tail, okay, and there is 5 UTR and third and UTR regions, okay, and these mRNA molecules actually synthesize inside the nucleus. So we don't find compartmentalization in case of prokaryotes. So hence the process of transcription and translation can take place at the same location, whereas in case of eukaryotes that is not possible. So this mRNA molecule formation will be taking place inside the nucleus, and that mRNA molecule will be transported to outside of the nucleus and in the cytoplasm of the eukaryotic cell, where the process of translation takes place, right? So this is about the monosystronic and polysystronic structural genes that we see, okay, like monosystronic in eukaryotes and polysystronic in prokaryotes. And now these mRNA molecules will be translated, and before translation we need to see genetic code. So the next concept I'll see about genetic code. So here it's a code that directs the sequence of amino acids in a protein synthesis. So it codes, okay, so it's a code that directs the sequence of amino acids, right? So here inside the cell, in the cytoplasm, we will be seeing a pool of amino acids, okay. Say I am talking in case of a eukaryotic cell, and in this from the nucleus the mRNA comes out here, and it will be having sequence of nitrogen bases, and these sequences of nitrogen bases will help to put these amino acids in a particular order to make a particular polypeptide chain. So these are all amino acids, and this is a nucleus, and this is mRNA. Right now there is a code that actually directs this sequence of amino acids. So what should be the next amino acid in the polypeptide chain? So that is all we will come to know after knowing this genetic code. Now we have 4 nitrogen bases. We have 4 nitrogen bases. Say suppose if one nitrogen base codes for one amino acid, if one nitrogen base codes for one amino acid, then in this case only 4 amino acids are coded. Only 4 amino acids are coded. So I'll tell you that the total number of amino acids that are used to synthesize polypeptide chains are 20. So there are 20 amino acids which will be used in synthesis of a polypeptide chain. Now if one nitrogen base codes for one polypeptide, one amino acid, then it can code for only 4 amino acids. And say suppose if 2 nitrogen bases code for one amino acid, then in this case it can code for only 16 amino acids. So that's you need to take something like this. It will be UA and UCAG. You can take some sequences, and again here UCAG, if you take like this, then total you will be getting 16 amino acids. And still these coding only 16 codes you will get, that codes for 16 amino acids. It means that this is also not correct. So we want for all the 20 amino acids the codes. Now it was actually a person named George Gamow. So this person, what he told is that as there are only 4 bases, and if they need to code for all the 20 amino acids, there must be a combination of these sequences, a combination of these nitrogen bases. And he proposed that to code for 20 amino acids there must be 3 nitrogen bases will be coding for one amino acid. So in that case, we will be having 4 into 4 into 4, so 64 codes we get, and they are called of course codons. So we get 64 codons, and 64 codons are more than enough to code these 20 amino acids, So that is a proposition given by George Gamow, and this is what we call it a triplet code. It's called triplet code. So in the beginning it's a singlet, and there is that is not correct, so this is not correct, and in case of doublet code this is also not correct. So we are not going to study about this. We will be seeing only about the triplet code, and of course later it was proved that 3 nitrogen bases will be coding for a particular amino acid. And the proof for triplet code, okay, that an amino acid is coded by 3 nitrogen bases. The proof for this, it has come from a scientist named Rajkumar. So from a person named Rajkumar, and he developed a chemical method, right, in which he synthesized some homopolymers and heteropolymers, right. So in this he synthesized something called homopolymers and copolymers, right. Of course, he synthesized RNA molecules. That is mRNA molecules with known sequences, right. So if I'm making a mRNA molecule, I will be knowing the sequences what are present. Say suppose A, G, G, like this. I know the sequences on the mRNA molecule. So that's what Rajkumar, he synthesized mRNA molecules, okay, with known sequences, and so they are like homopolymers and copolymers. In case of a homopolymer, uh, it will be having only one type of base. It will have only one type of base. So like you take all A are the sequences what you see here, right. So you will find right, so continuous, you will find same nitrogen base. It is then called homopolymer. So if it is the case, it's called homopolymer. And coming to copolymer, so these are actually heteropolymers, so where different nitrogen bases. So let's see, in case of copolymer, you may see sequences like UCC, again UCC, right, or we'll find a different combination. So in this case, I'm repeating those sequences. Okay, so this is a copolymer. It's a copolymer, right, with different sequences. And in case of homopolymer, you will see only one type of nitrogen base. Only one type of nitrogen base you will be seeing in this case. Okay, so copolymers and homopolymers. Now these sequences can be synthesized artificially, right. So it's called enzymatic synthesis of RNA, called enzymatic synthesis of RNA molecules. So enzymatic synthesis of RNA molecules, and this we can do it by using enzyme called Severo-Ogawa enzyme. So it's called Severo-Ogawa enzyme, and it's called actually polynucleotide phosphorylase is the name of the enzyme. So it's called polynucleotide phosphorylase enzyme, right. So we can synthesize these homopolymers or heteropolymers by using this enzyme called Severo-Ogawa enzyme, called polynucleotide phosphorylase. And this enzymatic synthesis of RNA will be template independent manner. Okay, so in this case, the synthesis of RNA will be in a template independent manner. will be in a template independent manner, means no need of template. So when we studied about transcription process, we know that one of the strand of the DNA will be acting as a template on which RNA synthesis takes place. But in this case, we need not require any template. That is the reason I use the word template independent manner, and I can synthesize a RNA molecule with my own sequences, means whatever the sequences I would like to see in a mRNA molecule, only that sequences I can, with that sequences I can make a RNA molecule, and by using this enzyme called polynucleotide phosphorylase, right? So Haragobind Khorana is a scientist who developed a chemical method where there is a synthesis of RNA molecules in template independent manner, and by using that enzyme called polynucleotide phosphorylase. And another scientist called Marshall Nirenberg. So another scientist called Marshall Nirenberg. So he developed a cell-free system. He developed a cell-free system for protein synthesis. We know that protein synthesis takes place inside the cell, but he developed a mechanism where the protein synthesis need not take place in the cytoplasm of the cell. So he constructed a cell-free system where these RNA molecules will be allowed to go for translation and result in the formation of a polypeptide. And now, finding out the amino acids in the polypeptide, say suppose I have a sequence, right, all the sequences on an mRNA molecule to be adenine, and then when I allow this to undergo translation, I get a polypeptide with only one type of amino acid, and that indicates that the AAA will code for that particular amino acid. Now we will see that. So once this all, so with all these cell-free systems and template independent manner synthesis of RNA molecules and all this has resulted in getting the genetic code, and this genetic code is triplet, right? So let's look at the genetic code. So we need to take 3 letters for every codon. So U, C, A, G, and again here U, C, A, G, and again this side U, C, A, G, U, C, A, G, then U, C, A, G, and U, C, A, G, right? And this will be the first letter of codon, will be the first letter of the codon, and this will be the second letter of codon, and this side it will be the third letter of codon, third letter of the codon. So let's see, right? So U, and this first one U, right? Next U, C, the next one U, A, and U, G, right? So these are the triplet codons what we get. The next one U, C, U, the next UCC, UCA, and UCG, right? So I finished the table. We can see that we have obtained 64 codons. So the total number of codons, So total codons we obtained are 64. So you can see here, right, each codon has 3 nitrogen bases. That is the reason why I mentioned it as a triplet codon. Right. Now the first one, UUU and UUC, this will be coding for, these two will be coding for an amino acid called phenylalanine. Okay, so here the PHE stands for phenylalanine. Right. The next one, UAA, UUG. So these two will be coding for leucine. So LEU stands for leucine. So the name of the amino acid leucine. The next one, all these will be coding for only one amino acid, that is called serine. SER, that stands for, okay, serine. The next one, UAU. So these two will be coding for amino acid called the tyrosine. So they code for TYR, that stands for tyrosine. Right. So 1, 2, 3, 4 tyrosine amino acid. The next below that, these two UAA, UAG, they don't code for any amino acid. Hence these two are the stop codons. So these two are stop codons. So they don't code for, right, any amino acids. The next one, these two will be coding for cysteine. So they code for cysteine. CYS stands for cysteine. Then below that, again we have is a stop codon. The UGA is a stop codon. It doesn't code for any amino acids, right? Then below that we have TRP. So this UGG codes for amino acid called TRP. So that stands for tryptophan. Stands for tryptophan amino acid. Next coming, okay, to this CUU and all these will be coding for only one amino acid. It is leucine, so which I already mentioned. The next one, all these will be coding for proline. So PRO, that stands for amino acid called proline. Then, right, so these two will be coding for histidine. HIS stands for histidine amino acid. Now below that we have is right, GLN. So this GLN will be coding for glutamine. So the amino acid called glutamine. And then all these will be coding for amino acid called ARG. The ARG stands for RG9. So ARG stands for, okay, RG9 amino acid, right? Then coming to this part, so here the first three codes or codons will be coding for ILE. So this ILE stands for isoleucine, and this AUG, it stands for an amino acid called MET. So that MET is methionine, stands for methionine. The next one, all these will be coding for amino acid called, right, threonine. THR stands for amino acid called threonine. The next one, okay, so here these two will be coding for ASN. Okay, that one stands for asparagine. That stands for, okay, asparagine amino acid. Then below this we have AA and AA, triple A and double AG. These two will be standing for or coding for amino acid called lysine. So LYS, okay, so that stands for lysine. Then in the next these two will be again coding for amino acid called serine, that I already mentioned, and below these two will be coding for ARG, arginine, which already I mentioned. Then coming to the last row, in this we will see all these will be coding for an amino acid called valine. VAL stands for valine. And then in this case these will be coding for an amino acid called alanine. ALA, that stands for amino acid called alanine. And the next one, these two will be coding for aspartic. ASP stands for aspartic acid. And below these two we have is, right, GLU, that stands for glutamic acid. So GLU is glutamic acid. And now here the last part of this genetic code, where we see all these codons will be coding only for one amino acid, that is glycine. So GLY is glycine. So you can see total we got 20 amino acids. Of course, the point is that one amino acid has more than one codon. Of course, later I will discuss about the properties of the genetic code, right? So out of all the total 64 codons, only 61 will be coding for amino acids. The rest of the three, that is UAA, UAG and UGA, will not be coding for any amino acid. So I hope earlier when I have drawn mRNAs in prokaryotes and eukaryotes, I have mentioned their start codon, okay, and a stop codon. So here this is the stop codons. So the process of protein synthesis will stop, right, if it comes across any of these codons like UAE, UAA, UAG or UGA. Right, with this I will end today's session. In the next session we will see the properties of genetic code, and the remaining process of translation. Will also start with the translation in the next session.